Welcome back. We are now moving into structure called lipid rafts. And probably you haven't heard of that before. There's a lot of introductory material in biology that kind of just doesn't focus at this level. But it turns out that there are specialized regions of our plasma membrane and other lipid uh, membranes that surround organelles that have specialized function and structure. They call these micro domains, and they're also known as lipid rafts here. So where you'd probably thought of kind of, if you think of the ocean as the plasma membrane, it's really the ocean with islands, and those islands are the lipid rafts. And they're very specialized, and that allows a compartmentalization of processes. And what it does, it allows the organization to occur at certain areas, so it's more efficient to have uh, the protein machinery, etc., signaling, fluidity, trafficking in one area or multiple areas associated with these lipid rafts. Lipid rafts are very ordered and more tightly packed and kind of float freely. So they're kind of like an island that's floating in the ocean, as it were. And there's also been lipid rafts reported in the Golgi and lysosomal organelles as well. So you've got the process, the activity of a cell is actually focused through these lipid rafts, and now that's putting them together, and that's where processes and signaling in and out of the cell can occur at these specialized areas. So it's probably more efficient when that occurs. So here's a cartoon showing a lipid raft surrounded by uh, the plasma membrane with its proteins, and they're specialized proteins and other lipids here. And so there's a special type of lipid called a sphingolipid. They are unsaturated and have fatty chains, and those sphingolipids have an attraction that is more attractive than phospholipids, and so that's what builds these lipid rafts here. Cholesterol, the reason you need some cholesterol in your diet, I mean, it's not all bad, is that they're also in these lipid rafts. And so you get sphingolipids, cholesterol, and specialized proteins that are all making and functioning in a so-called microdomain. And they're highly concentrated in cholesterol and sphingolipids. So if you could look across the plasma membrane, you would see these, the lipid and the cholesterol, the amount of those would change here. The lipid rafts, the fatty acids, the long hydrocarbon change of one layer actually interact with the other. So there is interactions holding together differential structure between the two leaves, whereas outside the lipid rafts that doesn't seem to be the case. And lipid rafts, these microdomains, will accommodate certain transmembrane proteins and lipid modified proteins as well. So there's proteins that are lipoproteins that seem to be found here. And so this little triangle here, this green triangle, okay, you see these little squiggles, those are actually the lipid attachments to these lipoproteins, and that's kind of like allows them to, kind of like if you put a candle in a birthday cake and that sits in the frosting. Well, these fatty acids anchor these lipoproteins into these lipid rafts. So you're getting a specialized structure that's building in these lipid rafts. Here's just a structure of the sphingosine, which is the molecule. There are a type of glycolipids that contain the amino alcohol sphingolene, and then there's three flavors of them, and you'll probably have to learn that in biochemistry. I just wanted to show you the structure, and these are different sugar structures, these neurominic acids, the nana sugar side chains are 60 gangliosides depending on the type of sugars, and it's thought to be involved in lipid rafts and modulates or helps in signaling transduction events. So they're finding, and of course, the cell has 60 known different flavors. So there's a lot of variation here. So some of these lipid sugar molecules that are attached to proteins have a specialized function, and it's called a glycophosphate inositol anchor. And so this glycophosphatidylinositol inositol structure, and it's shown here, there's a certain enzyme that will recognize a certain domain, and that is a product of the enzyme that will cleave, prolytically cleave this embedded region out and attach now this protein to the membrane through this GPI anchor, they call it. This is called a GPI anchor, glycosidyl phosphatidyl inositol, GPI. And so this is a hallmark of the proteins that are found in these lipid rafts. Not all proteins are GPI anchored, but a lot of them are found in these lipid rafts here. And this shows the process where that occurs. And so here's this lipid raft, and you can see the little orange structures. These are cholesterol. There's a bunch of those. There are glycolipids. Okay. There are proteins that bind glycolipids. They're called lectins. Here's a GPI anchored protein. Here's a transmembrane protein that has sugars attached to it. So see the specialized structure that's occurring in this lipid raft. You have specialized concentration of molecules that are binding on this side, the cellular cytoplasmic side of the lipid raft here. And so there's specialized structures and activities associated with them, and they're going to be very unique from the more traditional, what you've thought of is the lipid bilayer, which is probably like in this region, just phospholipids sitting there, very much not a lot of interaction, where there is a lot of interaction in a lipid raft. So they have a lot of sphingolipids, cholesterol, and glycolipids. And the raft can be on one side or the other or both. So some more variation there's no one size fits all here. This is just showing the cartoon, showing the structure here. And it can be asymmetric. So there tends to be specialized lipids. Here's phosphatidylcholine, sphingomyelin, and glycolipids for the plasma membrane. And the inside is sphingoserine, ethanolamine, and inositol. So they have found there's asymmetry, and that's part of making the structure here. And there's pretty much equal amounts of cholesterol here. And cholesterol makes this different fluidity than just the surrounding lipids there. So they tend to be extended, more tightly packed. They create these microdomains, and they float on a sea. If you think of an island floating on a sea of poorly ordered lipids, and that means a lot of these glycosphingolipids and other lipids are preferentially incorporated, and you kind of just form this lipid raft based on this order structure here. And unsaturated phospholipids with the bent tails will spread lipids out and make them more fluid. When cholesterol is there with bent and rigid steroid rings, they are more fluid but stiffer and tougher. And so the structure is actually much different than the surrounding unordered lipids. And again, they found cholesterol in proteins, and about 1988, there was a first discussion. But in the 50s, they would notice these structures called caveoli, and nobody really knew what they were. They looked like a little flask that was invaginated in the plasma membrane, and they were named caveoli. And now we know that caveoli are a type of lipid raft and a result of lipid rafts. So there's two flavors of lipid rafts: planar, which is more a flat lipid raft, so it's much harder to see on a plasma membrane, and the caveoli, which are going to be invaginated structures in a plasma membrane. So you have these more flat lipid rafts and these more caveoli invaginated structures here. And there's two flavors of the planar lipid rafts, the so-called glycosphingol enriched membranes, GEMs. And if there's not a lot of glycosphingolipids, they're said to be phosphoinositol rich rafts. So you have these inositol rich or these sphingolipid enriched rafts, but they're both look very flat, and they're called a plain or smooth lipid raft versus a completely different invaginated structure which has a protein known as caveolin. And planar rafts are defined as continuous. They're not invaginated, and you really can't see them, but they do have a specialized protein called flotilin. And when there's no caveolin, they tend to find these planar rafts with flotilin protein. And so caveoli are flask-shaped invaginations, and they contain the caveolin proteins, and they're easily seen and observed by their morphology. So planar rafts have flotilin, caveoli have caveolin protein, and they found these caveolins are in brain, in the nervous system, endothelial cells, a lot of nerve tissue. They both have lots of cholesterol and sphingolipids, and flotilin and caveolins can recruit into the lipid rafts, and there's a neurotransmitter signal transduction that's thought to be going involved in this structure here, and they have a lot of interactions that are basically needed for signal transduction. And we definitely want our nervous tissue to receive and generate signals. That's always a good thing. And so if you think of a lipid raft, this is how it would appear, kind of a floating island there, and stiffer because of the cholesterol. And again, the sphingomyelipids, the cholesterol, the gangliosides, the phosphatidylainositols, phosphatidylcholine, ethanolamine, serine are all different markers of lipid rafts here, and it turns out that lipid rafts are different. They can have different combinations of structures. In other words, a lipid raft is not the same. It's heterogeneous in structure. And there have been a lot of wonder and investigation into what little rafts do. Some are involved in cholesterol transport, movement of material in called endocytosis, and certainly signal transduction. Lipid rafts have been found to interact with the cytoskeleton. Actin binding proteins will bind to the phosphopolyphosphonosides and can be regulated. So there's a linkage between the cell structure and the lipid rafts there. But the planar rafts, the GEMs, are also linked to the lipid rafts through proteins that bind the cytoskeleton as well as the specialized lipids as well. And they're signaling platform for many receptor classes. This just shows a receptor here that's bound a target. So here the two monomers, when they bind, they actually come together and actually interact and form with a kinase activity. That's RTK, is receptor tyrosine kinase activity, and that ends up phosphorylating the cytoplasmic tail, which then attracts other proteins here. These little red dots are phosphate. So you bind the target, the receptors phosphorylate themselves, so they autophosphorylate, and then that attracts other proteins in or near the lipid raft, and that starts assembling a signaling complex that sends the signal towards the nucleus here. So they can up or down regulate receptors. And so the receptors tend to be near these rafts or in the rafts, so it makes it a faster. If you've got all your toys together, it's easy to build with your Tinker Toys if they're all in the box versus they're scattered throughout the yard. So a raft allows a lot of these players to be right there together, and so you go very quickly to this state once there's been a detection of some type of hormone or other signaling molecule from outside the cell, and you can start assembling these proteins together and start getting a lipid raft. So here are the caveoli, and they're different from the planar, which is the GEMs or the inositol enriched. Just showing there's a little different on how some of the references and how they may be isolated. The planars are also known as detergent resistant membranes, DRMs. So you might see that in the literature here. And a lot of them are definitely tied to signaling and maybe structure with the cytoskeleton. And there was 3 models of how lipid rafts are actually formed and what their nature is. And model 1 and 2 shows a process where we use 3 different types of detergent. And if the structure is layered, in other words, here's layer 1 and it's surrounded by, it's kind of like a cake, layer 1, 2, and 3. Then if you had a layered raft under certain detergents, such as the tween, you would end up getting the whole layer and it would look like that. If it was all these lipids together and the cake was all smashed together and all the layers were one, it would be a homogeneous raft. And when you did a a tween structure, you would end up getting this one structure with no layers. So the layered Ralph's model suggests it's this structure. The homogeneous Ralphs say it's this structure, and heterogeneous means that the Ralphs are in different uh, formations, different structures. And when you do the tween, you can find one, two, three, maybe even more types of lipid Ralphs. So these were the three models. So when they actually looked at all the purification, turns out that when they broke apart the proteins in the Ralphs, it's model 3, which is the heterogeneous, than either model 1 or 2. So it's the idea that lipid Ralphs are very different, and they're probably different in cells. They're probably different in individuals. Probably changes the efficiency of people's signaling depending on their genetics and what their lipid Ralphs are, how they're formed and what they're formed. I think it's kind of interesting that they'll start making some predictions. With failure, tissue or cells. So I expect that if that hadn't already been found, they will find something like that. And there's all sorts of different approaches. Easy, some are very hard. Here's our friend Fret. You can actually label multiple proteins and see when they come together in signaling. There's some different kinds of microscopy. Patch and visualize the proteins. Just to